Hi, I'm Dr. Ashley Ross. I'm a urologist and urologic oncologist at Northwestern Medicine. My specialty is the prostate and prostatic diseases, and in particular, prostate cancer. Recently, there's been a couple biomarker-driven clinical trials that showed very promising effects. Perhaps the most important biomarker-driven trial of late is a trial called the PROfound trial. That looked at men with castrate-resistant prostate cancer that had mutations in their DNA break repair machinery. The lead study investigator is actually Dr. Maha Hussain from Northwestern. And what she and the team showed is that for men who had mutations in their DNA break repair machinery, particularly mutations in BRCA2, that they would have a good response to PARP inhibitors. And if that was used as a therapeutic choice, then they would have an increased overall survival. And that trial led to an FDA approval of olaparib, which is a PARP inhibitor for use in men with castrate-resistant metastatic prostate cancer. A similar trial was done with another type of PARP inhibitor that confirmed these findings, and that was called Triton-2, and that also led to FDA approval of that drug, both of these in late May. And so now with a druggable target, it makes germline and somatic sequencing of tumors to look for these genetic abnormalities even more important. There are also many developing trials, some of them using, again, mutations in heritable DNA. One is looking at, can we expand therapeutic efficacy of immunotherapies in men with prostate cancer? We know that Keytruda or other checkpoint inhibitors have effects in men with mismatch repair deficiencies. There's also some newer data about CDK12, and if men have mutations in CDK12, a nice phase 2 study shows responsiveness to checkpoint inhibitors in that population. Finally, there's the concept of, we can screen genetics, somatic or germline, and identify some men that may be good candidates for drugs. But can we expand that out by looking at genomics, so expression levels as well? So Dr. Ted Schaefer, who's head of the urology department at Northwestern, has with other investigators developed an androgen response signature, and men with a low androgen response signature may be more susceptible to PARP inhibitors as well, and that concept is soon to be tested in a clinical trial initiating at the Lurie, hopefully in the next couple months. The utilization, however, of genetic testing and germline screening in men with prostate cancer is relatively low, and maybe that's because our understanding of precision medicine in prostate cancer has been relatively recent, or maybe because it hasn't had the same backing as some of the other cancers. So for example, when we think about BRCA2, which has a critical role in prostate cancer, we think about it in hereditary breast and ovarian cancer, and prostate cancer is not even mentioned in that. So one future is, how are we going to implement more widespread germline and somatic testing to identify who might be candidates for precision medicine? The second is, how can we boost what we're doing in precision medicine? How can we identify more people that are going to be responders to known therapies and help them with their therapeutic choice? This is happening in some of the clinical trials that are ongoing here at Northwestern or about to open here at Northwestern, looking at genomic and genetic signatures. And then finally, it's about developing newer inhibitors for other pathways that haven't yet been explored or have been somewhat explored in prostate cancer. A lot of prostate cancer will have alterations in the AKT-P10 pathway. There are new inhibitors being developed in that area. Dr. Vanderweel and others are going forward with clinical trials looking at those inhibitors for metastatic prostate cancer. There's alterations that happen with the regulation of chromatin also in prostate cancer, and there's newer EZH2 inhibitors that are also being investigated in prostate cancer. So really by encouraging us as clinicians to test for these genetic abnormalities, using gene panels that are broader in scope, selecting patients for clinical trials, trialing new inhibitors, and then in an iterative process, seeing who responds and who didn't respond, and can we understand along the non-responders why that was the case, and can we use different combinations of precision medicine tools for them? That's all going to push us forward.